Hello students, welcome to today's lesson on ecology and uses of bacteria. In the last class, we discussed classification of bacteria based on shapes and grams stain. Based on shapes, we categorized bacteria as cocci that exhibit spherical shapes, bacilli that show rod shapes, and spirochetes that have spiral or corkscrew shapes. We also categorized bacteria based on gram stain as gram-positive that has thick peptidoglycan and gram-negative that has thin peptidoglycan. We also saw the techniques to differentiate either a given bacterium is gram-positive or gram-negative. Now in this class, we are going to focus our attention on ecology and use of bacteria. When we see ecologies of bacteria, bacteria or prokaryotic cells or prokaryotic organisms are ubiquitous. That means they found every ecosystem. They can found in harsh or harmful environmental conditions. They can live on or inside any living organisms. In this case, they can live on or inside ourselves, our body. Since bacteria have different adaptational mechanisms, they live in all kinds of environments. They found in every parts of the earth, including ice-cold climate, hot springs, deserts, marshlands, and etc. What are the main benefits of bacteria? Bacteria provides many benefits or advantages to human beings. As we know, based on their values, bacteria can be grouped into two major groups, harmful and essential bacterial groups. Harmful bacterial groups are always known to cause disease, but essential groups of bacteria known to get applied in industrial processes and they involved in nutrient recycling in a given ecosystem. So what are the main advantages of bacteria? Let us see some in detail. Bacteria are important since even they cause disease. How they can benefit us since they cause disease? They can benefit us since they cause disease in two main ways. The first benefit is since they cause disease is knowledge and skill-based benefits, because the disease initiates human beings to do research and develop many knowledges about behavior of bacteria. This knowledge gives us the way how to prevent, control, and even eradicate this harmful microorganism or bacteria. The second benefit is since they cause disease are economical benefits. A country or a person who develops drugs and vaccines to pathogenic microbes or bacteria through research can gain high amount of money from the sale of the drugs and vaccines. The second main advantage of bacteria are their application in many industrial processes. Bacteria have been applied in traditional and modern industrial processes to produce certain valuable industrial products. For example, in traditional industrial processes, bacteria have been applied in alcoholic beverage production, in food production or food processing, in modern industrial process to produce fabrics, beverages, foods, and certain drugs and vaccines. The third main benefits of bacteria are recycling mineral substances in a given. As we know, nutrients are neither created nor destroyed. That means nutrients settled on Earth in finite amounts. So to keep the continuity of life on Earth, nutrients must be recycled in a given ecosystem. The role of bacteria and other microorganisms in infectious diseases. Before we are going to deal how bacteria cause diseases, particularly infectious diseases, first we need to define what is disease and health mean. Disease is a condition with a specific cause in which part or all of the body is made to function in a non-normal or less efficient manner. In contrast, health is a state of complete physical, mental, and social well-being. We will have more detailed information in the next class, but in this moment we need to talk about germ theory of disease. What is germ theory of disease, or what it claims? Germ theory of disease claimed or stated that infectious diseases are caused by pathogenic microorganisms. This infectious disease or communicable disease is simply a disease that's caused by pathogenic microorganism and that can be transmitted from one person to another person, from diseased host to healthy host. Who develops germ theory of diseases? Germ theory of disease mostly developed by three great scientists: Louis Pasteur, Joseph Lister, and Robert Koch. These three scientists cooperatively developed germ theory of disease. In the mid-19th century, Pasteur showed that microorganisms in the air caused wine to go sour. In 1860s, Joseph Lister also showed that carbolic acid or phenol acted as a disinfectant and prevented disease in bones following surgery. In 1880, Robert Koch identified Mycobacterium tuberculosis and Vibrio cholera as a causative agent to tuberculosis and cholera respectively. Then Robert Koch develops four postulations or four ideas. The four ideas or four postulations that Robert Koch put forwarded are: the first one, microorganisms must always be present when the infectious disease is present and should absent when infectious disease is absent. The second statement, microorganisms can be isolated from an infected person and then grown in laboratory on culture medium. The third postulation, what he put forwarded, is introducing such cultured microorganism into healthy host should result disease on the healthy host. The fourth postulation gives us the possibility to isolate microorganisms from this newly diseased host and culture them in culture medium in laboratory. These are the four main postulations that Robert Koch put forwarded. How microorganisms cause disease. In the last class, we mentioned groups of microorganisms based on their biological features. Now let us see how the four major groups of microorganisms, bacteria, viruses, fungi, and protozoa, cause disease. Bacteria cause diseases by producing toxins. Bacteria release toxins as they multiply. These toxins affect cells in the regions of the infection and sometimes in other regions of the body as well. Bacteria can also cause diseases through physical damaging the tissue or organs during invasion, because some bacteria invade and grow in the tissues of organs. During this process, they physically damage the tissue or organ. Bacterial diseases like pneumonia, cholera, pulmonary TB are some examples of bacterial disease. Second, how viruses cause diseases. Viruses cause diseases by disrupting the normal metabolic systems of the cell and instruct the cell to produce more viruses. You see, viruses enter living cells and disrupt the metabolic systems of the cell. The genetic material of the virus becomes incorporated with that of the cell and instruct the cell to produce more viruses. It makes the host cell as a machine to incubate their viral particles. Examples of viral diseases are influenza, HIV/AIDS, common cold, rabies, blah. The third groups of microorganisms, fungi, causes diseases by physically damaging the tissue. When fungi grow in or on living organisms, they grow their hyphae and secretes enzymes. This digests the substances in the tissue and the substances produced are absorbed. So the growth of the hyphae physically damage the tissue of the host. Some fungi also secrete toxins. These toxins, as we know, disrupt the normal physiological process of the host organism. Others can cause an allergic reaction. Examples of fungal diseases are athlete's foot, farmer's lung, and ringworms. The fourth groups of microorganisms that always known to cause disease are protozoa. Protozoa cause disease in many different ways, like by exploiting the nutrients of the host cell and expose the host cell to deficiency diseases. The second mechanism simply interrupt the normal activity of the host cell. Third, they physically damage the tissues of the host. It might be through bursting the host cell or in other ways. Examples of disease caused by protozoa are malaria and sleeping sickness. This is all over what I have on the ecology and use of bacteria. In the next class, we'll continue on how are disease-causing microorganisms transmitted.